So this semester at University of Glasgow has come to an end, and in these 3 months, 6 tigers and 31 leopards have been poached in India alone. These mysterious and majestic creatures who've once held high importance in religion and spirituality have been reduced to mere status symbols meant to be conquered. Well, today my purpose as an engineering student and as a STEM student isn't to bum you out or make you sad, but rather I want to explore how we could apply modern science and engineering to solve their conservation. How could we use historical evidence of evolution of these creatures and apply modern science and technology to protect their future? How could we use this knowledge to better understand our modern ecosystems and maintain their natural balance? And how much human interference is actually valid? Well, let's start by talking about what exactly is a big cat. Well, it's a colloquial term used to describe members of the genus Panthera. These include animals such as the tiger, the lion, the leopard, etc. And several non-pantherine felids such as the cougar or the cheetahs. Typically, they are large carnivores with specialized teeth, retractable claws, and function as ambush predators to hunt their prey. Now, big cats belong to what's known as a ghost lineage. A ghost lineage is basically a line of descendants descending from a line of lineage that we know exists, but we don't have lots of tangible proof for them. This occurs because fossils first off occur in very specific circumstances. And secondly, big cats are predators. Their populations will obviously be low compared to the prey specimens. When this happens, art is a good substitute as a secondary source when studying this. For instance, when you take the cave lion, which was an ancient relative of modern-day Panthera species, some of its most unique characteristics, like the fact it did not have a mane like modern-day lions, we only know this because cave paintings depict them as such. Now, when we study about the evolution of big cats, a good point to start is the Pleistocene, or the Ice Age, which is the age right before ours. Today, we call that the Holocene. So the one before that is the Pleistocene. We encountered large animals back then, which are referred to as megafauna. These include the Smilodon, the mammoths. And the reason why they were so big was explained due to the Bergman's rule, which states that animals that live in colder climate tend to have bigger body sizes to keep insulation. A good example of this is modern polar bears, which are some of the largest carnivores alive today. Another interesting fact is the iconic saber-tooth, which were present in many animals, such as the Smilodon and the Homotherium. However, they weren't as good as the teeth and the physiology of the Panthera species, which were a lot more versatile. The saber-tooths were actually, even though they look very menacing, they were very brittle and could be easily broken down. Well, an interesting question that arises here is, you see these massive beasts and you wonder, well, how exactly did they go extinct? We don't have a clear answer. It might be due to climate change, or it could be due to early humans. And if that was the case, if it was due to early humans, to wonder if this has caused an imbalance in the ecosystems. Every ecosystem, as you must know, has a specific niche for each organism to occupy. The plants are eaten by the herbivores are eaten by the carnivores, and so on and so forth. Now, it's suggested that during the end of the Pleistocene, several of these niches were left unoccupied and have caused an imbalance. Pleistocene rewilding is one such idea that aims to undo this imbalance by introducing similar species to fill these old unoccupied niches. For example, let's take the case of the American pronghorn, which evolved alongside the American cheetahs, which was an ancestor to a similar species to modern-day cheetahs. And because of this, they run very fast with a speed of 96 km/h. But unfortunately, with their extinction, in the entire continent of North America, there's no predator that can catch up to them anymore. And so Pleistocene rewilding would aim to reintroduce cheetahs back into this habitat so we can maintain these pronghorn populations. Another interesting concept floating around nowadays is that of de-extinction. I don't know if you guys have heard about this, but recently I was scrolling to Instagram and there was this post about bringing back mammoths. Could you raise your hand if anyone has seen that post? Yeah? So basically, that's what I'm going to discuss now. And to understand that, first we need to understand the science behind cloning, or somatic cell nuclear transfer. Just a basic biology recap. There are two types of cells in our bodies, the germ cells and the somatic cells. Germ cells are the sperm, the egg, and the somatic cells is everything else. When the sperm and the egg combine, they form a zygote. This is known as the stem cells. And we need to convert somatic cells to these stem cells, and this is where the principle of cloning lies. This is how we cloned the famous sheep Dolly that happened here in the Royal Institute in Edinburgh. And well, basically what we do is we take the nucleus out from the somatic cells and we introduce them into these egg cells. And then we electrocute them. This causes them to basically stop functioning as somatic cells and go back to this primordial stage of becoming stem cells. Then we introduce them into a donor, and the species that comes out, surprisingly, is not like the one that had the egg cell or the surrogate. It's from the one we took the somatic cell, and that's how we clone Dolly, the sheep. When we go into de-extinction, we just take this a bit further, and we basically what we do is we sequence the DNA of the animal we want to bring back. Say we want to bring back cave lions, which have gone extinct. We sequence the DNA of the cave lion and we compare it to modern day relatives. Say we take it compared to modern day lions. Then what we do is we introduce something known as the CRISPR-Cas9, which is a protein enzyme complex found within certain bacteria. And how that functions is it acts as genetic scissors. And we compared the parts of the two animals and what's unique to the modern lions, what's unique to the cave lions. We cut out the bits that make it into a modern lion and insert the bits that would make a cave lion. And now what's interesting here is DNA tries to close the gap between the cuts that we have created naturally. And we harness this natural process of closing the gaps. And then once we have that done, we just implement somatic cell nuclear transfer again. And theoretically, that's how you clone and bring back an extinct animal. That is what the mammoth cloning would essentially do. And once that, again, this is theoretically possible, but the main difficulty is sequencing the DNA, because till this date, we don't even have the entire sequence of humans. But that's how we can do it theoretically. Now, how do we further this? Well, what's interesting is we have something that's combining both these processes by a group of researchers here in Siberia, known as the Pleistocene Park, which is obviously it's a play on the Jurassic Park movie. So what happens here is they want to take the concept of Pleistocene rewilding and they want to introduce those animals back in this reserve in Siberia. And they've noticed something very interesting. When we go about conserving animals, we usually think about conserving the ecosystem, reversing climate change, and then we can save the animals. But what's happening here is we conserve these animals and they start to reverse the climate change themselves. So the park is divided into two places where you have these ancient animals and where you don't. And interestingly, the grass where you introduce these old animals back again, there's a lot more genetic diversity in them. Not only that, the ice there is colder because the way the animals moved, they trample on it, and the ice is held intact. That means the methane released into the atmosphere is also lowered. Again, another aspect that we could also do is take existing animals and just artificially enhance the versatility. Take modern day lions and make them more cold resistant so we can place them in northern habitats. Or we could take a tiger that could perhaps survive in Glasgow. And this way, we're just conserving them and just increasing their habitat. Finally, to conclude, and some ethical implications. Would people feel safe sharing these urban environments with these huge predators? Would you guys be comfortable coming down to University of Glasgow if you knew there was a pride of lions living in Kelvin Grove Park? And secondly, what's interesting is even if we do perfect cloning, these would still technically be genetically modified animals. They won't be an exact copy. Say you do reintroduce cave lions back into the ecosystem, they're still going to be raised by a surrogate modern day lion. They're still going to be raised alongside other modern lion cubs. They're still going to be eating the food that a modern lion would eat. And again, is there certain habitats, certain instincts, or perhaps behaviors that would perhaps forever lost to time? And finally, if we do manage to do de-extinction, wouldn't that drive away interest from actual conservation? Why would you want to invest in conserving a species when you know if that animal, say, for instance, does go extinct tomorrow, you could just bring it back by de-extinction as long as you have a surrogate species? Thank you. That's all for me.